Okay, so in this video, we're going to talk about the differentiation of the cells of the neural tube. So here we have a little cross-section of the spinal cord. And what we see here is three zones that this neural tube is divided into. So in regards to the spinal cord, we see, which right here, this is a cross-section of the spinal cord. It is made up of three regions, and those three regions are the ventricular zone, the mantle zone, and the marginal zone. The ventricular zone consists of stem cells, which are continuously dividing, and these dividing stem cells go on to form a second layer, which is known as the mantle zone. So here we have radial glial cells and neurons which are migrating to this zone here, which is the mantle region. Now, this mantle region is rich in neuronal bodies as well as glia, which branch out with axons, and they go on to form nerves. Now, this branching out of axons is what goes on to create a cluster of axons that gives us the marginal zone. It is known as the white matter area because these axons are wrapped in myelin sheath, and this myelin gives it that whitish appearance, and they're very poor in neuronal cell bodies. The mantle zone, however, it's mostly made up of neuronal cell bodies, and it lacks the myelin sheath, so therefore this is why it is known as the gray matter region. Okay, so moving on here to this region, which corresponds to the development of the cells in the cerebellum, we see that there are 5 zones which are comprised of the ventricular zone, the mantle zone, the internal granular layer, the marginal zone, and the external granular layer. So here what happens is that we have a number of stem cells that migrate all the way to the opposite pole, and these form the external granular layer. This external granular layer is highly folded, and it is composed of Purkinje neurons. And what these Purkinje neurons do is that they proliferate and they make contact with other cells that secrete BMP, bone morphogenic protein. The secretion of this BMP activates cells to become a different type of cell known as granule cells. And what these granule cells will do is that they will migrate back to the ventricular zone, but somewhere here along the middle, they will form this additional layer known as the internal granular layer. So back to the Purkinje neurons. These are very important because they offer a control where they provide functions such as balance. They relay information from the cerebellar region to other brain regions. And the major cell type that we find in the cerebellum is the Purkinje neuron. These Purkinje neurons, all right, so we see here this Purkinje neuron that dominates the cerebellum region. Okay, and what these Purkinje neurons do is that they secrete sonic Hedgehog. And what Sonic Hedgehog does in the whole process of the cerebellum cell differentiation process, it is that it sustains division of granule cell precursors in the external granular layer. Okay, so one very important concept that we need to understand about all of this, and we need to understand the difference between Sonic Hedgehog and BMP. So what BMP does is it actually gives the signal neural progenitor cells in the external granular layer to differentiate into granule cells. Whereas Sonic Hedgehog sustains the division of granule cell precursors. So Sonic Hedgehog aids in the division of these neural progenitor cells. So if we didn't have any Sonic Hedgehog, there would be no division of neural progenitor cells, which would therefore halt the manufacture of granule cells. Whereas if we had no BMP, we would have if we had no BMP with Sonic Hedgehog, we would have division of granule cell. We would have division of granule cell precursors, which are the neural progenitor cells. But with no BMP, we wouldn't get the granule cells. And without granule cells, we wouldn't get the internal granular layer. Okay, so lastly, we're going to take a look at organization in the cerebral region, which here is the cerebral cortex. And basically what happens is that it is organized in two distinct ways. Okay, first, like the cerebellum, similar, it is organized radially into layers that interact with one another. Okay, so certain neural progenitor cells from the mantle zone migrate on radial glial processes towards the outer surface of the brain and accumulate in a new layer, which is the cortical plate. This cortical plate will go on to form the neocortex. Okay, and this neocortex is a distinguishing feature in mammals. Okay, so there is a transcription factor, LHX2, which activates numerous cerebral genes and LHX2 provides a specification of the neocortex. The neocortex is stratified into 6 layers formed by neuronal cell bodies, where each layer differs by functional properties and the types of neurons that are found there, and also by the connections that they make. But we will talk more about these neocortex layers in a future video. So for now, we pretty much have an idea of how cell differentiation happens in the neural tube.